Welcome to Future Casting, a part of Ars Garden Leonardo Laser, a partnership with Ars Electronica and Leonardo. I'm your host, Pamela Winfrey. Welcome. Today we're going to explore the future, not the future of space travel or alien meetups or flying cars, but rather the future of a very specific biology, that of cancer, if you can believe it. I've invited three people who can help us explore this speculative terrain. Scientist Michael Metzger and two artists collectively known as Boredom Research. Dr. Michael Metzger, who was the first person to realize that certain bivalves were suffering from a type of transmissible cancer, was trained in the Pacific Northwest. He earned a master's degree in epidemiology and a PhD in molecular and cellular biology at the University of Washington. Michael's drive to further explore the evolution of this unique cancer and the resistance of the surviving clams led him to establish a one-of-a-kind lab at the Pacific Northwest Research Institute in 2018. Boredom Research is a collaboration between British artists Vicky Isley and Paul Smith. Inspired by natural environments, they collectively create artworks that explore the diversity that exists in nature and more recently, the fragility of these ecosystems. In 2019, the Arizona Cancer Evolution Center invited Boredom Research to become artists in residence. The resulting digital artwork, called In Search of Chemozoa, features a poetic rendering of an in silico model organism. This discussion compares speculative creatures such as chemozoa with actual real-life creatures. In discussing a speculative future, what other creatures could experience communicable cancers? And how do our imaginative creatures potentially inform our explorations in the future? So that's the topic for today. We thought we would start with Boredom Research because we are lensing this through primarily an art lens. So why don't you go ahead and take it away, Boredom Research.
Brilliant. Okay. Thanks, Pam. Thanks for the intro. So we're just going to share our screen now. And as Pam mentioned, collectively we work as Boredom Research and we create moving image artworks, often in collaboration with scientists, but also conservation groups. And in this really short 10-minute presentation, we're going to briefly talk about our collaboration with the cancer researchers at the Biodesign Institute. But also we're going to respond to the search for life before death, I think.
So we kind of start off with a disappearance, and this was something, it was an organism that was kind of observed by a German zoologist called Ernst Haeckel when he was looking for sponges off the Norwegian coast in 1869, and in one of his samples he came across this bizarre organism which he named the magician's sphere. And what he felt he observed was a kind of a ball of cells, a body of cells, and he thought that he saw cells leave the body and live independently. And to him, this was really kind of a profound moment, because what he thought he'd found was the missing link between unicellular and multicellular life. And this is still something that's a very kind of interesting subject in certain fields of science, and certainly in terms of cancer research, because it's kind of this key point between cells working together as a whole and cells kind of working independently and or maybe selfishly. But the thing was that this, true to its name, the magician's sphere, it kind of performed a bit of a disappearing act, and no one has ever seen this organism since. So it's something that kind of appeared in the scientific literature, but then has now kind of become a mythical being. As such. So, but like I said, it's this kind of connection between unicellular and multicellular life, or this idea of cells working together as a whole, which is very much how we think of ourselves in the human body, is as a cohesive whole. And when that breaks down, when cells start to act independently, that's something that we kind of refer to as cancer, or it's what we kind of consider as cancer. So, and also the interesting link is that in terms of evolution, that there's this kind of missing link between unicellular and multicellular life, and it's that same kind of transition that's very much the kind of the key aspect of cancer research, that transition from a kind of a multicellular cohesive whole to cells acting independently. And that's what brought us
so that's
to biodesign.
What brought us to work with the Arizona Cancer and Evolution Center. And that's where we met Dr. Angelo Fortunato, who is actually featured in our artwork in Search of Chemozoa. And we found Angelo interesting as he had a background not only in biomedical science, but also in ecology. So whilst we were in residence at Biodesign, we shadowed him in his daily work routine to understand a little bit more about his motivation and the methods that he was working with in the lab. And this is when we were introduced to one of the model organisms that he's working with, which is it's one of the simplest multicellular organisms called a placozoa. And they're very, very small. They're about like 1 millimeter in length. And they reproduce asexually. So all placozoa are genetically identical. And one of the reasons why Angelo is looking at this organism is there's been no reports of cancer in placozoa. So we found this really interesting because we could see the organism on a cellular level, but also as a whole organism as well. So we were really fascinated by this being. So Angelo spends a lot of time observing these placozoa, looking at them through a microscope, but there's still a lot of mystery around this organism, and that was another thing that really inspired us and gave us a bit of creative license as well to think about what these organisms could be like, other fictional organisms. So when we first met Angelo, the placozoa was one of a kind.
Literally one of a kind, isn't it? There was literally only one placozoa in the phylum Placozoa, which is quite exceptional. But by the time we finished, there were 3, so 2 more had been discovered in that period of time. It's about a 3-year project, wasn't it? So.
Yeah, but when we were in residence, it was about 1.5 years. Yeah. So it was kind of over that period that other placozoa was found, which is really interesting because obviously it's a new model organism. You're going to be learning things about it all the time.
And it really kind of drives home the kind of how much we don't know, how much life is kind of out there that has yet to be discovered. And that was really the central kind of theme of the work we did with Angelo, where we kind of just opened up this space of imagination and possibility and asked him to consider, other organisms that haven't yet been found and what mechanisms they might have or what behaviors they might have that would be interesting from the perspective of cancer research.
So very much inspired by his thoughts and the placozoa, we then conceived our own fictional organism, which is called the chemozoa, which you can see in the bottom here. And so this is an organism that was obviously yet to be discovered. And I think what we're going to do now is we're just going to play you a short trailer, which is basically, it features some clips from In Search of Chemozoa, the film, but also the documentary that we made alongside it, where we interviewed some of the scientists at the Biodesign Institute. It's very short. So.
Something had changed. There are a great many uncanny sounds below the water's surface, and now, among them, a new chorus.
So I think one vision of the future is that everybody will get cancer at some point in their lives, but you'll just live with it. And so it'll just be a normal part of a stage of life.
Angelo has managed to cultivate some amazing organisms in the lab. They're simple multicellularity. They don't have as many tissues as we do.
One thing we think about is evolution as a great problem solver, and then can we translate that into doing better cancer prevention in humans?
Why these animals, they are not developing cancer, and that they have something that we don't know.
A song made of a million frail voices united in a search.
Okay.
So we open the film as the clip that you saw right at the beginning is the opening of the film, where a camera is moving through an empty landscape among rocks that obscure our view. And in the film, we're literally searching, aren't we? And the narrator is introducing us to the idea that there's these mysterious beings that we are looking for and that we're searching for something. But there's also this other search that's going on, which is the search of the Chemozoa, isn't it?
Yeah. So I suppose in as briefly as I possibly can. So the Chemozoa literally have cancer built into the dynamic of their cells. So they're an organism that we kind of program that starts from a single cell, and as those cells divide, it has the mechanics of cancer built into that. And then in the film, we have a kind of a whole life cycle constructed around that being, where it's literally trying to manage its cancerous cells by establishing a balance between the aggressive mutant clones and the benign mutant clones. So it's very much kind of looking at a cancer from an ecological perspective and thinking about how it can balance that cellular environment to manage the cancerous progression. But then it does this through consuming toxic algae, and then in doing so, it's rebalancing the environment that organism is found in. So it kind of draws this kind of link between ideas of health at the level of the cell inside the body, but also the kind of actions of the body and how they impact on their environment, and how we can think about health both, across those scales, both inside the body and at the level of the environment.
And this was very much inspired by the research that we found during our residency, talking to the different scientists about what they're working on in regards to thinking about cancer and the balance of that, but also thinking about it like a sensitive ecosystem, and how we can think about these new sensibilities centered on balance. And I think we'll probably
I think
end there.
that's a good place to end.
Okay. So just stop sharing.
That's excellent. Boredom Research, as usual, you guys do an awesome presentation. So let's move on to the science end of this, and let's talk to Michael Metzger about what he's got going up in the Pacific Northwest.
Yeah, that's great. Thanks. That's really interesting to see. And I wanted to talk a little bit sort of about how I started working on transmissible cancer and then where we are now. And so initially, I want to say that the cancer is not normally contagious. Cancer is not normally transmissible. So what we're studying in these transmissible cancers are pretty rare events. So we often know of cancers in humans that can be caused by infectious agents. So there's a number of things that we know of, like HPV, a human papillomavirus. That's a virus that can transmit between people, and that can cause cancer. But when HPV causes cancer, and when other viruses or other infectious diseases cause cancer, what they do is they induce changes within each individual person's own cells. And so if a person gets cancer because of infection with HPV, those cancer cells will come from their own body, and those don't transmit from one person to another. If I have cancer cells, I cannot give those cancer cells to somebody else and have them survive. That just doesn't really happen. except in these rare examples. And so these rare examples, exceptions to those rules, those are the transmissible cancers. And before we started working on this, the two transmissible cancers that were known, one was in Tasmanian devils and one was in dogs. And the Tasmanian devils is the devil facial tumor disease. It's a solid tumor on the face of these devils, and it's actually transmitted physically where the cancer cells themselves go from one animal to the next when they bite each other, and cells will engraft from one animal into another as they fight and bite each other. And this is relatively recent. It's only been identified in the late 90s. And so it's thought to be a really relatively recent cancer. One of the unique things is that there's actually two separate lineages of that cancer that have appeared and are spreading through the population. So this is very rare, but it seems that when the conditions are right, it can happen multiple times. And so then the other transmissible cancer that was known before we started our work was the one in dogs. And this is a sexually transmitted solid cancer spread through sexual contact. And it's actually widespread around the world. And in contrast to the Tasmanian devil cancer, which is really recent, the genomic analysis of the canine transmissible venereal tumor, as it's called, predominantly by Liz Murchison's group in the UK, have shown that it is about 4,000 to 6,000 years old. And so what that means is that in this case of this transmissible cancer, the original founder animal that had this cancer lived 4,000 to 6,000 years ago and is long gone. And these cancer cells that came from that dog have basically been spreading as this transmissible cancer, sort of essentially as an asexual parasite through the whole population. And so it's basically been this unbroken lineage of living cells that really haven't been a dog for thousands of years. So it really does sort of get you to blur the definition of what an organism is and what we would call life. I think that's one of the really interesting parts that got highlighted in your In Search of Chemozoa piece. So I initially was not planning on studying this. So I did most of my training in virology when I was at the University of Washington. And when I was doing my postdoctoral research in the lab of Stephen Goff at Columbia University, I was supposed to be working on viruses. We were a virus lab, and there was an outbreak of cancer in clams that had been known for quite a while. And it was thought that it was caused by a virus, but people hadn't been able to identify the virus. So I, together with another postdoc in the lab, Gloria Arriagada, started working on these, trying to find the virus that was causing the cancer. And it turns out we found nothing. There was no virus. So our initial hypothesis was totally wrong. And when I started analyzing this further and trying to figure out what was wrong, we eventually came across the idea that it really, nothing else made sense. It had to be transmissible cancer. When we looked at the genotypes of the cancers looked like they were not the same individual that we found them in. And when we looked at cancers all along the East Coast, this was in a species called softshell clams, cancers from New York, Maine, Prince Edward Island, and Canada, they all looked nearly identical. They all looked like they came from the same individual. And so we found through that genomic analysis of the DNA of these cancers, that they had to have come through this transmissible cancer spreading. We've since looked at a number of other bivalve species. So these are things with bivalves, two shells, so clams, mussels, cockles. There's a large number of these species that have been studied by marine biologists, and they've found these cancers that are often called disseminated neoplasia or hemic neoplasia. And so far in every case we've looked where there is that cancer, we found that it is another lineage of transmissible cancer. So we now know that there's at least 6 reported transmissible cancer lineages in a variety of marine bivalves around the world. For the most part, they are within the same species they were found in. So the one in softshell clams stays within softshell clams. We've seen a few cases of cross-species transmission, but mostly within closely related species. So the mussels are really an exciting example that we started working on when I was postdoc, but then we developed this project in collaboration with a number of other awesome researchers. And we found that there is a single lineage of cancer that came from a species of mussel called Mytilus trosilis. And the same lineage of cancer is found in one species, Mytilus edulis, in Europe, and then another completely different species, Mytilus chilensis, in South America. So this cancer has spread literally worldwide from a single cell in a single cancer in a single individual many years ago to Europe, South America, and then more recently an independent group found that same exact lineage of cancer could be found in the Sea of Japan, just off the coast of Russia. So these things are really spreading all over the world. We're really excited in trying to understand how these cancers have evolved. And then the other side of it, which we think is really fascinating, is the fact that the clams are still out there, that the clams seem to be evolving resistance to these cancers. And we really are trying to figure out how these cancers are able to evolve resistance and how they're able to sort of survive in the face of this challenge of this sort of fatal transmissible cancer spreading through the population. And with the hopes that we can use that knowledge to understand sort of potentially new mechanisms to treat human cancers that nature has identified on its own in these extreme examples.
Wow, that's pretty amazing research you guys are doing. Let's move on to this idea of speculative futures. One of the things that struck me when you were talking, Michael, is that, you know, we're all looking at climate change as sort of this ever diminishing thing where pollution and things like that are really impacting the oceans in particular. Do you feel like that trend is going to mean that there's more cancers, not only in clams, but in maybe in other oceanic life forms? What do you think about that?
I think that there's a lot of interaction between genes and environment that's very clear, and especially when you start stressing organisms, their ability to defend themselves against pathogens of any number of types goes down. We've, in particular, we're starting to test whether or not these cancer cells are affected by temperature. And it does look, at least based on our preliminary results, like higher temperatures are better for the cancers, which is bad for the clams. But there's a whole other set of factors out there as well. And I think when animals are stressed, their ability to fight off pathogens of any number of types is going to be affected. So it's going to include these transmissible cancers. I think it'll probably make it worse, and I think it's going to be a broad problem.
I mean, do you feel like it's really interesting to think about chemozoa in terms of evolution? Would there ever be a chance that we would have a creature like chemozoa that actually has evolved to, I mean, you were talking about evolving resistance. Could it actually evolve to actually pull out toxins out of the water and be safe doing that? Is that even possible?
I don't know exactly how that would work. I think if the basic rule of evolution is whatever survives. It's sort of a tautology there when I try to think of how to find the basic parts of evolution. So if there is a way to get nutrients to survive that, then it will be selected for if it happens. So I think that just seems one of the things that we sort of learn, and maybe one of the things that comes out of this research, is that our definitions and our models of what makes an individual and what makes an organism, those aren't sort of hard and fast rules. Those are just sort of our understanding of how things work. And yeah, the nature and biology and disease is a lot messier than that, and it just basically follows the main rule of if it survives and keeps going, then it survives and keeps going. That's what it does.
And I suppose, like, if we're changing our environment so much, and like you know, like with nutrient runoff in water sources, with like things like harmful algal blooms and things like that, then there's species that are probably adapting to kind of like live within those environments as well.
Yeah,
right.
One of the questions I had, which is, well, the first question that you haven't, I mean, you've answered pretty much all the questions I had. You know, it's such a kind of a fantastic presentation in such a short period of time. But I mean, I imagine there's a big kind of interaction between the immune system in terms of the identification of cells that are foreign to the body, and that must be quite a significant thing. And I was thinking about autoimmune disease and how, you know, in general terms, there's a kind of an increase in both in autoimmune diseases or diseases that are kind of recognized as having an autoimmune kind of component to them. And and I think there's a lot of thinking that really is looking at that in terms of what is understood as a dysbiosis, that a disturbance to an organism's ability to, an organism's immune system's ability to correctly read its environment. Like in humans, the profound changes that we've had to our diet have created quite significant changes to our internal microflora, you know, microbiome, gut bacteria, and the kind of ecological structure of that. And that has kind of allowed certain kind of changes in, you know, because obviously that's the biggest interface between the immune system and the outside world in humans. I'm not sure how it works in clams. I suppose they're filter feeders, there must be kind of similar, but those disturbances literally kind of create a confusion to the immune system where it's unable to distinguish between, you know, it misidentifies self as a foreign kind of a foreign agent or a foreign body. And it seems like in this we have the kind of opposite thing happening, where it's not able to recognize a foreign body as foreign, and it's allowing kind of those cells to become part of an organism as opposed to being rejected. So yeah.
Right. So
Yeah.
we think about sort of our sort of non-self recognition of our immune system, that our immune system will, if a pathogen gets into us, it will recognize it as foreign and attack it. If we get the COVID vaccine, it will recognize that as foreign and it will attack it and protect us from significant disease there. Just a plug there. So if we, throughout our lives, we get exposed to a large number of different pathogens, and our body mounts an immune response because it recognizes them as not us. It recognizes them as different from us. The same sort of thing happens if we get cells from another person. So in a sense, the same sort of reason that I can't give you my kidney without immunosuppressive drugs, that's the same reason that I couldn't give you a cancer without immunosuppressive drugs, or you can't, or your immune system can fight off foreign pathogens and things. Your immune system recognizes anything that's not you and will try to reject it. And that's just, like you said, it's the main block to these transmissible cancers, is that your immune system will normally recognize it as foreign and reject it. And we know that in the dogs and devils, it's been able to evade that immune response. But being able to evade that is pretty rare, and we don't know exactly how it does that, but it sort of takes the mechanisms that your body uses to recognize cells as foreign and sort of prevents that specific recognition from occurring. And the groups that are working on that, that's a really interesting, outstanding question. How are those cases evading it? We can look at some of the examples. There's a few examples of small scale transmissible cancer in humans. So there's a few cases where people that get organ transplants and are on immunosuppressive drugs, there are cases where they can actually get an undiagnosed cancer from their donor. That's happened. So a pre-malignant cell from the donor will sort of sneak in through that organ transplant, and you can get cancers growing up there. And so like you said, that if you evade the immune system, then you can get these transmissible cancers. And we think about all of these immune responses as just sort of a given for organisms, but it's only actually in vertebrates. All invertebrates, they do not have the same sort of non-self-recognition system we have. So clams and anything else that's an invertebrate, they don't have those same mechanisms. And so that we think that's why that the bivalves are sort of uniquely susceptible to transmissible cancers, or more susceptible to them than humans are. That said, we still sort of have invertebrates, and so we don't know, there must be other mechanisms that they have that we don't know about that can prevent this kind of cancer. And like you said, the immune system is a double-edged sword. You can fight off pathogens, you can fight off cells from other organisms, but you also have the autoimmune disease, and that is a balance. And if you think evolutionarily, sort of, there's going to be some tuning of that as to if you decrease your immune response too much, then you won't get a response to pathogens, and if you increase it too much, then you'll get too much autoimmune disease. And it's sort of that is a difficult balance for to make, and yeah, that gets to a very complicated question. There's a whole bunch of questions about diversity in the population and sort of different genes that can affect this, as well as like you said, massive changes in our environment compared to when we were evolving as a species, and that's definitely going to affect it.
I mean, one of the questions that non-scientists will probably ask, so I'm going to ask it, is I'm assuming that no matter how many bivalves we consume, we would never get cancer through that, right?
Yes, that is right.
Never evolve to get cancer like that, right?
Right. So so we will not. We are totally safe from the clam cancers.
Just had to say that.
Yeah.
I think it's a good question.
Yes.
It was the first thing that when we mentioned your research to the curator that we're working with here, it was the first thing that she
said, I love seafood.
I love shellfish.
Yeah, yeah. I love raw oysters, so I would be really heartbroken if I had to give that up. So we're almost
there's
out
no
of...
thought.
Oh, good.
You're totally safe to eat oysters. I probably ate, well, there's other things like but those are the
like parasites and stuff.
Yeah. But the cancer itself.
Thank God.
The cancer itself will not affect you. So like it said, there are transmissible cancers in different species. The cancer from softshell clams can't be transplanted into the mussels or vice versa. So even a clam is safe from mussel cancer, mussels are safe from clam cancer. We're totally safe from all of them.
I'm sure if you're a clam, you're very happy to hear that. Are there any last thoughts before we wrap this up? This has been a great conversation.
Yeah, I mean, I suppose my kind of last question is, I mean, from a scientist's perspective, and because I think I'm wondering how much you think in terms of I mean, you're kind of privileged in the way of kind of thinking about these kind of cellular behaviors and life at the level of those kind of cellular behaviors, and how much that's kind of impacted your own kind of maybe self-identity. You know, humans are just we just very naturally think of ourselves as a single being. And I think in terms of coping with, or you know, being able to kind of really engage with ideas of climate change or climate crisis and ecological collapse, and really kind of think about the part that we play in that. There's a kind of a different way of thinking that's much more fluid in terms of that way of thinking in quite a selfish way, that's kind of cancerous in, you know, in its own way, rather than kind of thinking in a more kind of collectivistic way. And I was just wondering whether your kind of research had kind of given you any kind of insights that you kind of respond to as a human, you know, in terms of your everyday kind of everyday kind of human behavior.
Yeah, and I think one of the things you're getting at, that you're piece highlighted as well, is that there's sort of this, the way that people and animals evolve in their environment. You can think of cancer evolving in their environment, sort of evolving as an organism. So a lot of the same concepts and the same types of interplay will occur at all these different levels. And then, like you said, at the level of ecology, you can have these interactions occurring. I think it's interesting to think along the lines of what Athena Ectyptus talks a lot about, in terms of cancer is sort of a cheating cell that is cheating in the game of multicellular cooperation. So in order for multicellular organisms like us to even exist, all these cells have to be working together. And when the cells cheat and go out for their own selfish purposes and divide, that turns into cancer. And for the most part, it's a dead end. It's an evolutionary process that wins in the short term, but it's a dead end. And you can think about our behavior in the larger society, and there's definitely analogies to, are we working together? Are we cooperating together for the good of the unit of larger orders of people, or are we sort of acting as the cancer and taking the short-term profit and potentially destroying the larger organism and the larger community for those short-term profits? So it sort of lets you think about those different themes and those different concepts at different levels, and that they all seem to be pointing towards cooperation as a good idea if you want your host system to keep functioning.
I think that's actually quite a lovely place to stop. It's a little bit more positive than some of the things that we've been talking about. If you like this podcast, please feel free to share it on Instagram and Twitter and Facebook friends and things like that. And I wanted to thank Ars Electronica and Leonardo and Michael and Paul and Vicky. Thank you so much, and tune in for the next Leonardo. Take care, you guys. And thanks again for being willing to come and talk about these things. Really appreciate it.
Thank you.
It was great fun. Thanks.
Yeah, thank
Bye.
Fun talking to you.
Bye.
Bye.